Hello students, today's topic is integrons. So let's discuss about what are integrons. Integrons as a mobile DNA elements. So before that, let us discuss about the transposable elements in short first. So transposable elements in bacteria contain one or more genes not related to the transposition that can also be mobilized along with the transposons. This type of element is called as transposons. Okay, so here the transposable element in bacteria that contain one or more another genes related to the transposition that can also be mobilized or transposed along with the transposons. Okay, so these elements are also called as transposons or the mobile DNA elements. So transposons can incorporate into plasmids where they can be transferred to the recipient cells by the process of conjugation. You might have known about the process, that is the gene transfer between the bacterial cell, that is the gene transfer between cells by forming a bridge like structure that is also called as pilus. Okay, so this process is the conjugation process in bacteria that may also transfer the transposons, or that is also responsible for the transposition mechanism. Okay, so transposable elements are flanked by inverted repeats and often contain multiple antibiotic resistance genes. So this is the characteristics of transposable elements or the DNA element, that are having a transposable element that is flanked by inverted repeats, that are right inverted repeats or the left inverted repeat at both the ends, and they often contain multiple antibiotic resistance gene in between the genome, okay, or within the genome. Integrons are the DNA elements that encode site-specific recombinase as well as a recognition region that allows other sequences with similar recognition regions to be incorporated into the integron by the recombination. So in the integron structure, it is having a specific gene, that is the site-specific recombinase gene, that produces the site-specific recombinase enzyme. Then as well as it is having a recognition site that allows other sequences with similar recognition regions to be incorporated into the integron by the process of recombination. Okay, means that recombination or the recognition region is nothing but the ATTI site. Okay, that we'll discuss with the help of figure later. Okay, so integrons are mainly having the site specific recombinase enzyme first, and the second a recognition region. Okay, so integrons are the elements that the elements that integrons acquire are known as cassettes. Okay, so this gene cassettes is a circular antibiotic resistance coding region flanked by the recognition region. Okay, so this gene cassettes additionally an integron will usually contain one or more gene cassettes that have been incorporated into the integrons. Okay, means at a time one or more gene cassettes that can be incorporated into the integron structures. Then these gene cassettes may encode gene for antibiotic resistance, although most genes in integrons are characterized, okay, or maybe they are uncharacterized. Okay, so this an ATT sequence, that is the cassettes are having, are also called as 59BE, is present that flanks the cassettes and enables cassette to be integrated at the ATTI site, that is the main recombination site, that we'll see with the help of figure, and then it get excised and undergo horizontal gene transfer. Okay, so the gene transfer in integrons is with the help of horizontal gene transfer mechanism, and that is the site specific recombination. Okay, so the integron may acquire multiple antibiotic resistance cassette which results in the plasmid resistance to the large number of completely unrelated antibiotics. Means the bacteria acquire this multi-drug resistance character by the incorporation of these gene cassettes into the integrons here. Then the bacteria with resistance to multiple antibiotics are an increasing problem in public health and particularly among the gram-negative bacterial pathogens. Okay, so the bacteria with the resistance to antibiotics are increasing day by day now. That the integrons may be found as a part of mobile DNA elements such as plasmids and transposons, and they can also found in chromosomes. So integrons are the common structure in plasmids, transposon and the chromosomes as well as. Okay, so the discovery of integrons were initially discussed on the conjugative plasmid through their role in antibiotic resistance. Indeed, these mobile introns or the integrons, sorry, not introns, mobile integrons as they are now known can carry variety of cassettes containing genes that are almost exclusively related to the antibiotic resistance. And further studies have come to conclusion that the integrons are chromosomal elements and their mobilization onto plasmids has been fostered by the transposons and selected by the intensive use of antibiotics. So the discovery of transposons initially they were the conjugative plasmid through their role of antibiotic resistance. So the function of majority of cassettes found in chromosomal integrons remain unknown, but researchers are doing work on it. So the structure of integron is minimally composed of first a gene encoding for a site-specific recombinase, that is the int-I gene that belonging to the integrase family. So the most important part of the integron is the site-specific recombinase gene that is called as the int-I gene, which produces the site-specific recombinase enzyme for the site-specific recombination or the horizontal gene transfer. Then second, main recombination site is the ATTI site that we have just discussed before, which is recognized by the integrase enzyme and at which the gene cassettes may be inserted. Okay. So this ATTI site is the main recombination site for the incorporation of gene cassettes. They are shortly called as GCs, and at a time two or more gene cassettes may get incorporated into this ATTI recombination sites. Then the PC promoter, the promoter which directs the transcription of cassette encoded gene. So this promoter is for the transcription or which shows the direction of transcription of cassettes encoded within the genes. Then the multiple antibiotic resistance units can be assembled to form integrons. Okay, so the multiple antibiotic resistance character of gene cassettes, it get inserted into the integrons. So here in this figure you can clearly see the function of integrons and its transposition or the incorporation of the GCs, that is the gene cassettes. Okay, so the integrons may be found as a part of mobile genetic elements such as plasmids and transposons and chromosomes also. We have just discussed about this before. So the terminology of super integron was first applied in 1998, but without the definition to the integron with a long cassette array on the small chromosome of Vibrio cholerae. So in 1998 the first study observed in the Vibrio cholerae bacteria. Okay, that is, the term has since been used for the integrons for various cassettes array length or for the integrons on the bacterial chromosomes, that is for example plasmids, use the super integron is now discouraged since its meaning is unclear today. Okay, so this figure is showing the incorporation or the site-specific recombination of gene cassettes in the integron. Okay, here you can see in this figure int I gene, that is the integrase gene, in blue color. Then you can see the PC promoter, and in light green color you can see the recombination site, that is the ATTI site. And GC1 is the gene cassette 1, then again ATT 1, then the GC2, that is the gene cassette 2, which are get incorporated into this integron structure. Now the crossing between the ATTI site and the GC3, that is the gene cassette 3, which is indicated by the red color here. Okay, so this crossing indicates the site specific recombination between the ATTI site and the ATTC3 gene of the gene cassette. Okay, so this is the main antibiotic resistance character of the gene cassette. Here the incorporation of GC3 you can observe in the second figure, that is the GC3 is get incorporated by the site specific recombination or the horizontal gene transfer. You can see here the GC3 is get incorporated into this integron structure. Then before that GC1 and GC2. And here in the third figure you can see the removal of GC1, that is the gene cassettes, from out of the integron structure. And the remaining GC3 and GC2 you can see in the last figure. Okay, so here the acquisition of cassettes make possible transcription of protein coding sequences. Okay, but here cassettes lack its own promoter. Okay, that's why it use the PC promoter for the transcription, because they are lacking its own promoter. Gene cassettes are lacking its own promoter. Okay. So now significance of transposons. The transposable elements T are DNA sequences that are capable of integrating into the genome at a new site within the cell of its origin. Okay, so this is the peculiarity of the transposon, that is capable of integrating itself into the new site at any site within the cells of its origin. Okay, that is the main function of the transposon. Sometimes the change in their position creates or reverses the mutations, thereby altering the cell's genotype. So this is the main significance of transposon, that it reverses mutation or alters the cell's genotype. Okay. Then secondly, the transposable element that are also called TEs, transposons, jumping gene or the junk DNA, anything you can use for the transposable mobile DNA element, is a DNA sequence that changes its position within genome, right? Sometimes creating or reverting the mutations and altering the cell's genetic identity and genome size also. As you know, the mechanism of transposition, it can be cut paste or copy paste. So due to this copy paste mechanism or the cut paste mechanism, it alters the cell's genetic makeup, and so that with the help of copy and paste mechanism, it increases the genome size. Okay. Then the transposition often results in duplication of the same genetic material. Okay, as it get copy and paste within the genome, so that results in the duplication of the same genetic material. So the ability of transposons to increase genetic diversity. So this is again main significance of the transposable elements, that it increase the genetic diversity among the species, or the together with the ability of genome to inhibit the most transposable elements activity, which results in a balance that makes transposable elements as an important part of evolution and gene regulation in all organisms that carry this sequences. So this transposable element play major role in the evolution and the gene regulation of organisms. Okay. Then transposable elements can also cause multiple changes in gene composition and function. So the gene composition also may get changed because of these transposable elements, and function of the particular gene may also get changed by the incorporation of these transposable elements into the functional gene. Okay, means this, if the transposable element get incorporated into any particular functional gene, it can lose its activity. Okay, so that it is responsible for the change in the composition and function also. So these changes range from the simple insertional mutations to the genetic reprogramming, gene movement, and the evolution of the novel functional protein coding sequences. So this is again the main important significance of the transposable elements. Then the transposons play an important role in the evolution and the diversification of the centromeres also among the closely related species. Okay. So here they are also playing an important role in the evolution as the diversification of chromosomes is occur. So the changes caused by the transposable elements are potentially rich source of variation on which the selection can operate. So again, the transposable elements play important role in the evolution or the variation on the selection of species. Then given large number of plant transposable element on evidence for ongoing activity of these selfish genetic elements, it is reasonable hypothesis that the transposable elements have played an important part in plant adaptation also. As we have already discussed in the first lecture about the transposable element in plant, that is the transposable elements in maize or the maize, that are the AC and DS element, that are the dissociator and the activator factors in the maize, that are responsible for the purple color or the colorless kernels in the maize. Okay. So almost half of our DNA sequences are made up of transposons. Okay. So they jump within the genome in developing sperm and egg cells and are important to the evolution. So they jump within the germ line or into the developing sperm and eggs so that it carry out the evolution in the species. But their mobilization can also cause new mutation that leads to the diseases such as hemophilia and cancer. Okay. So this transposition, it may be harmful, that causes the fatal diseases like hemophilia and cancer also. Okay. So here that's all about the integrons and the significance of chromosome. I hope you like the lecture and you understand the integrons better. In the next video, we'll discuss about the another transposable elements. Thank you.